I would like to thank you, Dr. Joannès Guillemot, very much for accepting our invitation to join the TropiBEF workshop 2020 and to give us a talk about the MataDiv project. Dr. Joannès Guillemot obtained his PhD in plant ecophysiology from the University of Paris-Sud and AgroParisTech in 2015. His research includes forest ecophysiology, functional ecology, and biogeochemical cycling in forest ecosystems. Currently, Joannès Guillemot is a visiting professor at the University of São Paulo, ESALQ. He is one of the coordinators of the MataDiv project, which is a tree diversity experiment that aims to improve our understanding of functioning of restored forest in the Brazilian Atlantic forest. Welcome, Dr. Joannès Guillemot. The stage is yours.
Thank you very much, and thank you very much for your invitation. It's a great pleasure for me to be here and maybe to share the story beyond MataDiv and who knows the mistakes we made in the process. Maybe could be interesting now that we are discussing the beginning of your great experiment. So yeah, this MataDiv project is the result of a collaboration between CIRAD, which is a French research institute, and the University of São Paulo. So we are basically three co-PIs, three friends: Pedro Brancalion from the University of São Paulo, Agnès Robin and myself, who are from CIRAD, but also at the moment based at the University of São Paulo. And do I have the control of my presentation or not? Yes. So we basically share exactly the same context of the BEF experiments. This is why we very warmly welcome this initiative, and we're very happy now to share very little experience of this young experiment. MataDiv is also based in the Brazilian Atlantic forests, relatively close to the BEF Atlantic experiment in the state of São Paulo, close to the city of Piracicaba. So I just checked, it is still 8 hours driving from the BEF Atlantic experiment, but let's say that in terms of the Brazilian standards, it's relatively close. And the idea behind MataDiv is to explore the effect of tree diversity on forest functioning, obviously, on the ecosystem services provided by the forest, with two specific objectives. One is to provide theoretical new knowledge on the mechanistical link between biodiversity and ecosystem functioning. And the other is really to give practical knowledge that could possibly guide the restoration projects that are ongoing in the region. So I guess we share a lot of your objectives too. I mean, the objectives of the BEF Atlantic initiative. One strong focus that we had in mind since the beginning is that obviously the mechanistical link between diversity and ecosystem functioning is strongly relying on complex feedback between the soil compartment and the trees. This is one scheme I made when we wrote our first proposal to fund the MataDiv experiment. And it shows that obviously the feedback loops between soil and trees are fundamental, or are likely to be very important in a number of contexts. This is why we wanted to build an experiment. Sorry, I'm a bit, I don't know what the control of my slides is a bit messy. We wanted this experiment to allow for some tests of these loops between trees and soil. So, yeah, and we wanted also this MataDiv experiment to be a long-term multidisciplinary platform aiming to host and welcome a number of collaboration. This is also why we are very interested in this Brazilian BEF network that is being built now. So the MataDiv experiment is set in the one of the field stations of the University of São Paulo, which is very interesting in terms of facilities. We have field lab, we have everything for the student and for the professor to stay several days in the field. And in terms of the perennity of the experiment, it is also very interesting. The whole area is 30 hectares, but the MataDiv experiment itself is 8 hectares. It is here. So located in the state of São Paulo, just a bit north of the BEF-Atlantic experiment. We have 4 blocks that are distributed parallel to the topographic lines. There's not big slope, but anyway, we distributed the blocks parallel to this topographic line. And within each block, we have 36 experimental plots that I will now explain a bit further, for a total of 144 experimental plots. So the idea beyond the BEF experiment that we set up was to cross several factors. So obviously tree diversity, I will now explain this a bit further, but also other factors that are fertilization, nutrient fertilization, because this is very important this is a very important management practice in the restoration project, and we wanted to manipulate it to see whether or not it impacts the ecosystem functioning, in what way, and how it interacts with tree diversity. And another factor we wanted to manipulate is the water availability, like mimicking climate change or the higher intensity of drought expected under climate change. So we partly also cross a fruitful exclusion with tree diversity and with fertilization. So beginning by tree diversity, our main species pool includes a number of 6 species. We have 6 monoculture, 4 mixtures of 3 species, 1 mixture with the 6 main species, and 1 control modality with 20 species. We do not have all the monoculture for this 20 species mixture, but this is somehow our control. And we follow the broken stick design a bit like they did in BEF China too, to ensure that all species are equally represented at each diversity level. Now I will go back to this slide. Because we wanted, so the whole experiment have been implemented in a former eucalyptus coppice. As you can see on this picture, the eucalyptus are being harvested. It was some months before the implementation of the experiment. So one first difficulty we had was to convince the harvester to get the trees out of the parcel on the day of the cutting, because we wanted to manipulate fertilization and we wanted to avoid nutrient leaking from the trees to the soil after harvesting. So you can see, like I compare in this picture, normal harvesting, where the branches are kept on the ground, and on the right, MataDiv, where the soil is clean. It's clean because we wanted to have an effect of this fertilization manipulation. The 2 treatments of fertilization we used are very contrasting. I won't go into detail, but I just wanted to give you the figure here. One is very close to what is used in the fertilization, in the restoration project of the region, and one is very, very reduced. It's absolutely limiting the growth of the trees. It is meant only to avoid mortality at the beginning of the experiment. Species selection was also a very important decision, at least as relates to our 6 main species. So at the beginning, we wanted to base our choice on a panel of functional traits in order to be sure to have a complementarity of ecological strategies. But it turns out to be very difficult to gather all these data sets before implementing the experiment. So we finally based our choice on a little number of very important criteria that were a large distribution of each species, as you can see here, a large difference in the ecological strategy of the species, at least in terms of position in the succession and in terms of potential growth. And other criteria that were much more practical, this availability in nurseries, for example, because we planted 30 hectares with mostly 6 species, so we needed a huge number of trees at the same time available to be planted. It was not so easy after all, so we had a lot of practical difficulties like this. So our choices finally include these 5 species that are dicotyledon. WD is wood density, so you can see that we aim to encompass a huge gradient of wood density, which hopefully corresponds to huge differences in terms of ecological strategy and potential growth. This is also very coherent with what we already know from this species. And we included the palm tree, because we know that palm trees are very important for the dynamic of the forest in tropical biomes, and very little is known on their effects in the diversity effect in tropical forests. And we included Fabaceae, but that is not fixing the atmospheric nitrogen. This was also a very debated question, but finally we decided against choosing a fixing species. The main reason was that we wanted to avoid the pitfall that would be trying to answer all the questions with only one experiment. So because we are manipulating nutrients, nitrogen, phosphorus, and potassium, we decided against using in this experiment a species fixing nitrogen to avoid mixturing the effects. So this is an overview of the experiment. You can see that the through-fall exclusion in red here, the red frames, have been gathered. So the overall design is close to a split plot design. This was done for practical reasons, because if you want to really have an effect of the exclusion, you need to concentrate the gutter in the same area. And it's also much more convenient to do, because the whole area of gutters is 2 hectares. So it is absolutely huge. So we needed to gather this area. Planting pattern, very similar to BEF Atlantic. So the same tree spacing, 2.3 in hedgerows, 2. So we have 100 trees per plot, with 36 central trees in the part of the plot that will be measured effectively. And we also use an intra-plot planting pattern that is based on a semi-constrained algorithm that maximizes species interaction, but introduce some random sampling in the planting. So the monitoring of MataDiv will focus on carbon, water, and nutrient cycling. And as I said, we'll also focus strongly on tree-soil interaction. So we plan to measure on a regular basis fine root dynamics, carbon fluxes, including soil respiration, tree growth, obviously, water fluxes with soil water dynamic, tree transpiration, with subflow derived estimates, tree resource use efficiency, especially light use efficiency using a modeling approach, and nutrient use efficiency. Soil microbial community with a focus on mycorrhiza, and microclimatic variables that are likely to link soil process and tree functioning, such as, for example, soil temperature. Some pictures of the experiments. An important point, as you can see here on the right, was that we managed to have a high tension, high voltage line reaching the experiment, which may appear like a detail, but if you want to monitor continuously carbon or water variables, this is actually fundamental, because maybe 80% of the problem you get usually comes from power supply. So this we managed this because of the strong support of the University of São Paulo, and this was really a great achievement, and it meant a lot to us. Some problem we ran into during the establishment of the experiment that was led in 2019. We had a strong grasshopper attack on one species, on the mutambo, and we reacted a bit late. So now the mutambo, that is supposed to be one of our high growth potential species, is actually very small compared to its growth potential. So this was one of the difficulties we had. We had the problem of phytotoxicity using one pre-emergent herbicide that is commonly used in the station, but some species reacted very badly to it. Didn't kill the trees, but it meant a delay in the growth. So this was also something we learned in the process, that not all species react the same, and you cannot, if you didn't grow these species specifically, you cannot so much rely on your experience from the past. So these are problems we could have been avoided. And now we also had a drought problem, which was a bit out of our hand, but as you can see here from the series, we faced one of the worst droughts in the last rainy season, and this led to some mortality event in our experiment. But we decided to replant the trees that died at this occasion, because we considered that mortality at this very early stage has nothing to do with an ecological process. So we decided to just give them one full rainy season to grow, and then replant once, and then to start monitoring mortality from this point on. Pictures recent of the experiments are Cecropia species, which is the species with the highest growth potential, is now 2 meters high, 2.5 meters, and the other species are between 50 centimeters and 1 meter. The first inventory is then planned in April 2021. So this is also, it means that we will start to monitor these sites, which is a very good news, the first data. And then the fruitful removal, the fruitful dispositive, which is not installed yet, will be installed at the end of the next rainy season, so in late 2021. These are pictures from another fruitful exclusion experiment that we manage in the same field station, so it will look exactly like this. But we wanted to have 2 rainy seasons full for the trees to grow before installing this exclusion. Okay, almost finished. I just wanted to mention that MataDiv, as I said, is included in a much larger area of 30 hectares, as you can see here. And these 30 hectares has been planted with the 6 species that are being studied in MataDiv. So we have a huge area of planting with the same age and the same 6 mixture planting that we have in MataDiv. So we took advantage of this and installed very close to the experiments an observatory site for the monitoring of carbon, water, and energy cycling. We have an eddy covariance flux tower, humidity soil sensor down to 6 meters, soil temperature, soil respiration, and so on. So there's a strong connection between these sites because obviously these are the same species with the same management, but these observatory sites aim to capture the fluxes at the ecosystem level, and this will add on to the dataset we will gather in MataDiv. Exactly in the same way, we also cross a third experiment called Matadam, because we set up different treatment of the 6 species mixture, manipulating restoration practice. So the control of grass with glyphosate, with mechanical control, with permanent vegetal cover, using another product, indisiflam, that is supposed to reduce the amount of chemical product that you put in your ecosystem. And the idea is to analyze the effect of this practice on soil, vegetation, and runoff water, to evaluate the impact of restoration practices on ecosystem functioning. So just to mention, it's a bit out of the focus of MataDiv, but this is all linked. And this is one of the great advantage of this site, to cross different theoretical and practical questions in the same area, I think. I just, to finish, because I think I'm running late, I just wanted to tell you that what we did before cutting the eucalypt, so before installing the experiment, was to characterize the soil, what we call T0. We characterize thoroughly the soil before tree cutting. As you can see, this is the eucalyptus coppice. So we characterize all the plots, the 144 plots being sampled in surface, 0 to 10 centimeters, and in depth for some plots down to 4 meters. And we characterize the physical chemistry, the functional diversity of microbial community, the microbial abundance of microbial communities, and the diversity of microbial communities, encompassing bacteria, fungus, and mycorrhiza. This was the great work of Agnès Robin, who could explain this in much more detail than myself. But yeah, I won't go into detail there, but just to say that the idea was obviously to have a clear view of soil functioning before installing this experiment, but sampling the exact location of the parcel that has been planted. So these are preliminary results. I don't want to explain them now, but I just wanted to mention that the idea is obviously to go back there in some years, sample the same parcels, and really isolate a tree diversity or tree composition effect on soil functioning. In terms of the scientific perspective, we had a project funded by Brazil and the European Union in the Biodiversa Code that has been recently accepted, and MataDiv is one of the sites that will be explored in this project, aiming to explore the potential of mixed plantation for climate change mitigation and adaptation. And we have we have some other projects submitted. This one that aims to explore the effect of nitrogen and phosphorus nutrition on the functioning of tropical plantation and how it interacts with tree diversity. I mention it because it's very much in the focus of soil-tree interaction that we aim to continue in the future. So this is it. Again, Leonardo, Gustavo, thank you very much for your invitation. I very much hope we are going to start a long-term collaboration between experiment and beyond. And if you want more information, please contact us. And for now, if I'm not running too late, I'm very happy to answer any questions.